Okay, then let's get going. Some more, well, it's sort of not gene technology directly in terms of the manipulation of the genes, but sort of some of the apparatus and techniques that goes around it, some PCR, some electrophoresis, and maybe a bit of a summary of the gene tech that we've done so far. Okay, so uh, let me know first of all how many of you are in the exambulance. I started this with a similar little session for the year 12s just now. If you want to get inside the exambulance, then I think the link's in the description below. So taylorstutors.co.uk, and you will be able to go and sign up to the exambulance. To basically, it gives you structure for your studying at home. It's there to support you. And also, yeah, do I have any other questions for you right now? For you year 13s, we are going to be making an announcement on Friday about how the grades are going to be awarded to year 13s, about your calculated grades. We haven't set a time for it yet. It kind of depends. We will tell you as soon as we are allowed to tell you. It's not been publicly confirmed yet. So we will basically give you an update. So follow the YouTube channel. We will schedule a live event for Friday afternoon, and we'll let you know sort of as soon as we can, if we can. The live classes on YouTube are going to be replaced by the live classes inside of the exambulance itself. So if you want to continue joining these live classes next week, make sure in the exambulance, we're going to be creating a page in there with the schedule so you can see what's happening and when. And that way we're going to do them inside of a Zoom room. So you will need to download Zoom, the application, and we will be doing live classes inside of there instead of doing them on YouTube, just because it's clunky doing them on YouTube, to be honest with you. Okay. Note that the exambulance is going to be free. There will be probably additional live class. I mean, our paid subscribers get additional time with us. Obviously, they can ask us questions, and like any of they get access to the full content guide, the full exam guide. That everything's exam board specific. I don't have to sort of mince my words about editing content for who's doing which exam board, because the courses are all fully exam board specific. I'm going to be doing less teaching next week. I will be doing more support with you, drop in questions. So if you've got exam paper questions, we're going to be guiding you through our TT revision method. So keeping you sort of sharp on all the different topics. So lots of different bits and pieces, but they will be, available. You won't need to register. You'll just need to be inside the study at home exambulance to join us on the times. I'll be doing them in the afternoon. I've responded to your feedback. You're saying it's a bit early in the day for you. So I'm going to be doing mine. I think I'll be doing mine 2 till 3 in the afternoon, every day. That's the plan right now. Again, we're changing it. We're responding to what you want, what we can do, what's going to be most useful. So if you're watching the recording for this sometime in the future, jump into the Study at Home exambulance. It's free, and we'll be there doing live classes. But who knows what structure or form it will have taken on by then. We might be doing something totally different. How I would like to know, in fact, a quick question to each of you before I get too stuck in. I want to drink a sip of my tea. How did you find Tailored Tutors in the first place? Where was it? I'm actually going to leave it as an open-ended question. How did you find Tailored Tutors? YouTube's responding pretty quickly today. That was only like 3 seconds for you to give me some answers. So YouTube, friends, and student room. So the half-term pass that we did, YouTube. And what do you think it is that makes Tailored Tutors different from other online study resources? Super responsive. YouTube was like this all the time. We could do our live lessons on YouTube. It's easy to understand, it's more personal. Notes templates are really useful, specific and straight to the point, precise, concise. See improvements in a short time, or it does really make a difference to it. That's what we focus on, efficacy. Yeah. Quality of the teaching makes sense. You get your questions answered. It's not a lot of jabber, and it's example specific, true. Okay, cool. Well, we talked about PCR in a fair bit of detail the other day, but I'm going to go over some of the basic notes on PCR. I've moved my iPad to a new position today, so I'm going to see how this goes. So what is PCR? What does it stand for? What is it used for? I touched upon this in terms of, we talked about how they're testing for viral RNA. If you have the coronavirus, if it's SARS-CoV-2 virus inside of you, how do they do that? Well, you basically take a sample that contains loads of DNA, lots of different organisms' DNA or RNA, and how do you only copy the bits that you want? Well, it's all about using the right primers, basically. Amplify DNA fragments. Okay, perfect. So polymerase chain reaction, and you're making many copies, millions of copies in a relatively short space of time of DNA. So let me share my screen. I've figured out how to share my screen without a cable as well. I'm very, very pleased with myself. Oh, but I do need to open AirPlay. Share this. Done. Look at that. So quick. Yay. You can see what I'm doing. Okay, I'm going to try writing my notes up here, which is going to be kind of weird for me, but it hopefully means I can look at the camera a little bit better. So I'm going to zoom in just a little bit to make writing notes a bit easier. So it's used to amplify DNA. Which means make, I'm going to say millions of copies. Sometimes, obviously, from a crime scene, you might only get a few fragments, or from the swab of somebody's throat, they might only have a little bit of DNA. It can be called in vitro DNA amplification. So in vitro means in a test tube, and DNA amplification. So it's just another way of describing it. I'm going to say can be called in vitro DNA. That's not what to use, just want to recognize if they were talking about that kind of in glass is what vitrio or vitreous means glassware. So basically in the lab. The ingredients is what I put here, obviously not the most scientific language, but it's what do you put in the pot that we start with in terms of making PCR happen. We're going to be making DNA. So we need some building blocks for DNA in there, right? So what do we need to put in the pot? Is PCR one where isolated genes are used? Typically, no. Typically, you're isolating a gene so that you can put it in somewhere else. So I'd say it's probably not one of those. It's mostly used for DNA fingerprinting or profiling, or anywhere where you've got a small amount of DNA and need more of it. So you're going to need the DNA sample, which is the thing that you want to make copies of, I suppose. So let's start with a DNA sample. You're going to need DNA nucleotides. Probably worth specifying DNA nucleotides. I just did my AS session on transcription translation, so they're not RNA nucleotides. So I'm going to call them free DNA nucleotides. So what is a nucleotide? We've got our phosphate, our deoxyribose, and then our nitrogenous base. I'm going to represent this as a, if this was an A, then I'm going to represent this as a little T shape like this, because we basically form, if we're drawing DNA, we draw it like this. It's just way simpler to represent those nucleotides in that fashion. Yeah, we've got our DNA nucleotides. What else have we got in there? We've got, so some people have put promoters, Katie, and some people have put primers, so BAPE. So what's the difference here? What's the difference between a primer and a promoter? Why do we not use nitrogenous bases instead of the whole nucleotide? Well, with the bases, to form the DNA needs to have the sugar phosphate backbone. So we have our base like this. These are the nitrogenous bases, but they wouldn't be joined together. You can't make DNA without the backbone, which is this bit here. So you need to have the nucleotide. The only thing that's unique about the nucleotide is the actual nitrogenous base, though. That's why often you only sort of represent the base, because the base can represent the whole nucleotide. That kind of makes sense. The promoter is the region immediately before the gene on the DNA strand. And the primer is the bit that's complementary sequence to the promoter region. So in the DNA sample will contain the promoter, and this will contain the primer, which I will define as a, I don't know, short sequence of DNA, where it's the DNA that is complementary to the promoter region. So it's the sequence of DNA complementary to the promoter region on the DNA. We're also, basically this is, I'm going to say this is used to select which DNA is amplified. And the enzyme, what enzyme do we need? The primer is a sequence of bases, not just a base. You can name it specifically as Taq polymerase. I would, Nima, are you OCR? In fact, to be honest, no, the enzyme you use in here is going to be DNA polymerase. It is Taq polymerase typically, because it's, your OCR may, I mean, they don't ask it as a factual detail. It could possibly crop up in those multiple choice questions because they ask all kinds of crazy questions in those multiple choice, but I would universally OCR and everybody, I would say it's DNA polymerase. Taq polymerase, I'll come on to the details about the enzyme a little bit more. Ligase, what's ligase used to do? Ligase is used to join. You've got this bit of DNA here, and you want to connect it to this bit of DNA here. You're going to be joining together. In this circumstance, well, we're not going to be doing that. So we don't need DNA ligase in this circumstance. Okay, let me draw it and then we'll see. So we've got, I suppose actually, I'm going to try and do something that I've not done before here. Let's see how this goes. This is my, in my pot, in the beginning, stage 1. We've got my double-stranded DNA base. I am going to be modifying my diagram in a way that I have not done before. This is going to be my promoter region. This is the DNA that I specifically want. Let's say there's contamination in here for other DNA. I'm trying to decide what color I want to do it in. It's not going to mess with my diagram later on. I'm going to do it in purple because I don't know. I haven't used purple on my other diagram. So let's say there's other DNA in here as well from, let's say this is from a dog. This is DNA from a cat, just contamination. This is kind of a bit of a, and this is the human being that we want to copy the DNA. This is the DNA in black that we want to make a copy of. And the red bit here is our promoter region, which is the sequence immediately before the gene. Occasionally, you might not be copying genes, you might be just copying DNA in general, but we're not going to go down that road for right now. So I'll maybe put, this is the promoter. So the first thing we need to do, whilst we've got our bases here, we've got all our hydrogen bonding going on between our double-stranded DNA. It's all paired up. Our A's and C's and T's and G's are all attached to one another. Same with obviously these over here, but we're not basically interested in the dog and the cat DNA. I'm just putting that in there because it's going to help us understand the use of the primers. What do we do first of all? Step one, we're going to heat to 95 degrees C. Why do we do that? We're going to break the hydrogen bonds to make the DNA single-stranded. I'm just, yeah, okay, so Katie, you're quite right. It's not always a gene that we want to copy. Sometimes the DNA fingerprinting or profiling is done with the non-coding sections of DNA. So the primer can technically be a bit of DNA that's complementary to the beginning of the sequence that we want to copy, which might not necessarily be coding DNA, but you're quite right, yeah. So the promoter regions will be complementary to one another. So you are quite right. They won't, because of the anti-parallel nature of DNA. If this is C, oh, I can't write that small, C, G, oh, I'm going to avoid using G's, C, C, T, A, then this is obviously going to be the opposite. They will be complementary to one another. That is quite correct. So we'll need two types of primer because one is going to have to be complementary to each strand. Okay, so that's stage one. We're going to heat the DNA, make it single-stranded. In fact, let me make my number one over here the same color so that it matches number one. All the DNA is going to become single-stranded at this point. I'm only really going to draw I've got my top strand over here. How do I know it's the top strand? I'm just gonna okay, well, I've got my promoter region, so I'll keep my promoter region just so we know which way around it is. I think it's going to be easiest. Oh gosh, making this diagram more complicated is potential problems. So this is the top piece of the DNA. And then we've got this piece over here, and then we've got the bottom piece of the DNA, which has got my promoter region like this. So we've got our sort of unpaired hydrogen bonds here. And this would have also happened for we've got our single-stranded bits of dog and cat DNA as well. But what do we do next? We've heated it, we've got single-stranded DNA. We're going to let it cool to around about 50 degrees. They'll give you a little range of these. I'd say a bit cooler than 70. 70 is one of the temperatures we need, but not here. This is, and why do we do that? The reason is allows the primers to bind. The proper word is anneal. You could just say bind. This happens by complementary base pairing. Again, you want to be using, every time you've got two bases joining up together, A with T or C with G, use the phrase complementary base pairing. They're the marks every time. So this means that the beginning of the DNA is double-stranded. So DNA polymerase can bind. Can. Let's say only bind when the DNA is double-stranded. Okay, so let's draw that over here. Step 2. Actually, I can probably stick with this. Yeah, I think I probably should draw my bases in here. So we've got what color did I draw my primers in? I drew my primers in blue. So this primer is going to be this primer is going to be so these are my primers, my short little sequences. I don't draw them as 3 bases typically because it's too easy to confuse them as triplets and it's got really nothing to do with triplets. And this is where it's kind of interesting with our other purple DNA over here. Obviously, they don't have this sequence. So the primers do not bind to any of the other contaminated DNA that's in there. So here I just did some dog and cat, but it could be other human, it could be whatever. Like there's no match. We could maybe say the primers do not bind because they're not present. It's actually right there in black. So primers do not anneal, let's say anneal. And it has to be cooled down. Let's put 95 degrees C on here, and we can put, in fact, let's put it next to the number. It's going to be a bit easier. Here we've got about 50 degrees C. And it allows, if it's too hot, they're vibrating too much, it basically melts, it breaks those hydrogen bonds. So here we can maybe say the tick, tick, and obviously because it's different DNA, it's not, we haven't put the primer in, we choose which primer goes in. How long are the primers? Not long, up to 10s, I would say. If someone wants to Google that, then they're welcome to let us know. I don't know, I'd say 5 to 20 bases, but I could be quite wrong with that. Pretty sure they're not long. It'd probably be easier to Google how long is a promoter region of a gene. Doesn't need to be much, but now we can basically, now that we have the double-stranded DNA, our enzyme, DNA polymerase, can attach. Whereas over here, the primer does not anneal, and therefore the DNA polymerase is not attached. Do you see how, therefore, by choosing which primers we put in, we can determine which bits of DNA is copied? 18 to 22, boom, nailed it. That is not. when it's inverted, well, previously, Ruhab, we had the top strand and the bottom strand. So I'll put T and B, top and bottom. This is our top strand. Now they're single stranded. We've got the top and the bottom. This is the top and this is the bottom strand. Oh, interestingly, no. Yeah, we don't need to know in any more detail than this. I think there's actually a slight error in my diagram, the way I've drawn it here, but it's not a problem. The enzyme is DNA polymerase. Maybe we could label that. And obviously what it's going to do now is do its thing. Now that it's attached, it can do its thing. It can run along and add on both of these strands. It can add free nucleotide bases so that we end up with, let's draw it there. And then for this guy, well, this is where the diagram is slightly wrong, but I'm not going to dwell on it right now. No, I selected blue quite clearly. And then our enzyme is here working its way along. It's going to be adding in the next base, basically. Same as DNA replication at this point. Can you see the error in my drawing? Oh, actually, it might not be a technical error. It might actually happen like this. I don't really know, and you definitely don't need to know. And then we've obviously got our sort of spare nucleotides are just sort of hanging around, waiting to be added on one at a time. And that's why it's DNA polymerase and not DNA ligase, because you're adding one nucleotide after the other, which is the role of DNA polymerase. If you were joining two long lengths of DNA, then you'd be using DNA ligase. Okay, so we're going to complete this until we basically end up with two completed strands. So what are we actually saying? This is actually stage 3 that we're talking about right here. So notes for stage 3, we're going to reheat it. What's the next temperature? Heat to around about 70 degrees. I generally go fairly round numbers for this because they don't make it that specific. Well, okay, I guess I'm going to join these down here. I'm going to say heat to 70 degrees. DNA polymerase adds complementary nucleotides to form phosphodiester bonds. By forming and it obviously reaches, and then you're going to have two double-stranded sequences. You're going to finish this with one double strand here and one double strand here, and we don't copy the other DNA that was in there, the contaminated DNA, because there was no primer for it. It wasn't double-stranded at the beginning of it, therefore DNA polymerase couldn't attach. So it doesn't matter what goes into your sample. It can be a whole mixture of DNA. You can choose which bits you copy by just adding the primers complementary to the beginning of the bit of DNA that you want to copy. And then basically the fourth step would be repeat. Now we started with one strand, one's double-stranded strand. Now we've got two double-stranded strands. So each cycle we double. So repeat stage 1 plus 2. Oh, I'm going to zoom in a little bit. The stop recording, the stop screen sharing button is right next to where I'm writing right now on my screen. It's kind of hard. So repeat stages 1, 2, and 3. And then maybe I'm just going to put here each cycle doubles the DNA, because a single strand is made into, one double strand is made into two double strands. Each cycle doubles the DNA quantity. So it goes 1 to 2 to 4 to 8. It doesn't take long to get into many cycles. Depends on how much DNA you need. Usually you would end up with millions, and it doesn't take that long. Okay, so why doesn't it take that long? I suppose we've gone from 1 to 2, and we'll go from 2 becomes how is it so fast? What's unusual about the temperatures? And this links back to Taq polymerase. Did anyone notice the to what extent diagrams is accepted as answers? I sometimes would use a diagram. You'd have to annotate it. A diagram with no annotations, no marks ever. But a fully annotated diagram, labeling the enzymes, labeling the strands, labeling what's happening, is credit worthy where relevant. Transcription, PCR, these types of processes, it could be mark worthy for sure. High temperatures, 72 degrees. Yeah, so I've said 70. I think 72 degrees or 74 degrees is what they actually run it at. What's weird? How can you, what's DNA polymerase doing at 72 degrees? What would happen if I took your cell and I put it in a water bath at 72 degrees? Would denature. So that's why people are mentioning Taq polymerase. Taq polymerase would be added from the beginning. It would just replace the DNA. Taq polymerase is a version of DNA polymerase taken from a thermophilic bacteria. Thermophilic, aquatic. What does the Q stand for? Who knows what I can't remember what it stands for. And you don't even need to know Taq polymerase. You can just call it DNA polymerase. But it's extracted from a thermophilic bacteria that has adapted to live in hydrothermal vents, and therefore it can withstand heat. But because there's more kinetic energy, the reaction can happen much faster. So that brings down each cycle time, which means that we can run these things much faster. Yeah, from an extremophile, a thermophilic bacteria. Yeah, maybe what I can do is can I pause my screen sharing? I think I can pause it on here. Yeah, it's because of this program. Oh no, I'm seeing. I have no idea what you're seeing. Are you seeing the Edexcel spec now? Or are you seeing my notes on PCR? Okay. Excellent. You are still seeing that. Okay, so over here, to summarize, we start off with one double-stranded bit of DNA. We're going to basically, step one, we're going to heat it to 95 degrees C. It's going to make it really, going to melt it basically. And now I'm going to draw the top strand here with unpaired bases kind of coming down like this. And then the bottom strand is over here. So they were like this, they've melted and they've come apart from one another. So the second step is cool to around about 50 degrees. And that allows our primers to bind. I did the promoter region in red on my other diagram. I don't tend to draw, it's 3 bases just so it doesn't confuse it as a triplet. So this allows the primers to bind to the beginning bit of the gene. If we wanted to, we could color this bit red. And only the DNA that's got the bit that's complementary to the primer will this happen to. Any other contamination DNA is just going to be single-stranded. It's just going to sit there doing nothing at this stage. Now that it's cool enough, DNA polymerase is going to bind on to each strand. There's going to be multiple copies of a DNA polymerase. That DNA polymerase is technically called Taq polymerase. This is all done by hydrogen bonding in here. And there's going to be 3 DNA nucleotides. So if this is CGAT, the first base that we want to add on is, well, I've not really got enough space for it, but it's going to be partnering with this C. We're going to have a G come along. And if I were to zoom, I can actually probably zoom in a little bit here. Well, actually, I'm just going to draw my G in there, and I'm going to draw the C that's attaching next. And the DNA polymerase, in fact, let's get rid of DNA polymerase here. I'm going to draw it slightly differently so that it's a bit easier for me to draw. So if we're adding on our C here, then the DNA polymerase is catalyzing the reaction that joins the phosphodiester bond, which is this reaction here. And the next one that's coming in to pair with the A, it's not partnered yet. This would be our T nucleotide, our thymine nucleotide, just lining itself up. And again, they're going to be lined up by hydrogen bonding, 3 hydrogen bonds between C and G. There'll be 2 hydrogen bonds between this and the T when it comes in here. But this one would not be connected yet because my DNA polymerase is still here. It hasn't got to this bond yet. Basically, that's then going to continue until we end up with 2 double strands. So before we had 1 double strand, and now we've got 2 double strands, 1 here and 1 here. So every cycle we double the DNA. And then what do? Well, we just repeat. So we heat it, and now we have our 4 single-stranded pieces, and then we repeat on loop. Step 2, primers anneal, and because of complementary base pairing, and that makes the DNA double-stranded at the beginning, which means only DNA polymerase can only attach where the DNA is double-stranded. Yeah, we can do a quick question on this. Let me find one. The easiest for me to find it. Okay. I'll open all of them. I wonder if I can do it in here. Let's try it. It's a mark scheme, it's a mark scheme. It's not quite right as a question. Okay, it's an application question. It's not. It's more of an inheritance question than it is a PCR question. It's the context is PCR. Let me check another one here. It'd be nice to find one's a bit more straightforward. Okay, I think this will do. Let's try this. Let's share. Oh, I love this new screen share. Amazing. He says with a black screen. I'm assuming you're also seeing a black screen. Literally as a, are you gonna do anything for me? No. Try again. So I'm lucky. Okay. I'm assuming you can now see that. Some populations of flies, let's choose a pen here, are becoming resistant to insecticides intended to kill them. So we've got resistance to insecticide, and we're talking about flies, and they want to kill them. Scientists developed a method for finding out whether a fly was carrying a recessive allele R, little r, that gives resistance. So little r is the resistance to the dominant allele, which does not give resistance. Okay, so just which way around are they? Crossed flies with the genotype big R with a little r. This is the typical place you're going to start a breeding experiment. Homozygous dominant with homozygous negative. They obtained DNA samples from the parents and the offspring. They used the same restriction endonuclease enzymes on each sample to obtain the DNA fragments. Okay, so it's the same restriction endonuclease. Explain or give reasons why the scientists used the same restriction endonuclease enzyme on each DNA sample. We don't know what they're doing with the DNA here. We're not inserting it into a target gene as far as we know. What are we doing with it? We are so they're obtaining samples and they want the samples presumably to include the genes or the alleles for the gene R. You type. So to produce sticky ends. So sticky ends are mostly going to be used when we're inserting them again. This is a tricky one. I'm not 100% sure. You want to cut out the same sequence here because we want to compare this bit of the genome with the same bit of the genome in another organism. So typically, we're talking about sticky ends being the same because the enzyme is the same. I don't know how relevant that is in this particular question. Do you see why that might not be the case? I'm just trying to think. I'm trying to think and talk at the same time, which generally leads to bad thinking on my part. But I would say so that it cuts the DNA at the same locations so that you isolate the same sequence of bases so that you get the same bit of DNA extracted in the different organisms. I wouldn't necessarily contaminated DNA. I will check the mark scheme. My answer would be something along the lines of it cuts at the same sequence or recognition site. So you extract the same region of DNA so that it contains the R/R gene for comparison, It contains the same loci. In fact, I don't need to put /gene loci is a great thing to say. Do you see how I don't think, I might be wrong, I don't think we're not putting this gene into something else at this point. The only information we have is that we're comparing DNA. I want to compare this bit of DNA from myself with this bit of DNA from another organism, and therefore I need to cut the same bits out, therefore I need to use the same scissors, basically. Okay. The scientists added 2 different primers, so 2 primers to each sample of DNA fragments for the PCR. Primer A3 only binds to a 195 base pair fragment from allele. Okay, so we don't, the number of bases, it might be useful, but the allele little r. A4 binds to big R. Scientists separated the DNA fragments produced by the PCR on a gel, so it's gel electrophoresis, where shorter fragments move further. So this is the direction, so they're moving in this direction. So the shorter fragments, which is this big R or little r, question mark here. Okay, the smaller one, yeah, moves further. So this is the big R and this is the little r. So all of that information is available to us in the question stem. So what's the genotype of this person? What's the genotype of L, M, and N? So give me L equals. L has only got 1 band. This tells us nothing about how much DNA is there. Oh, in fact, in this case, they have done it quantitatively. Normally, you wouldn't get that. You see how this is basically twice as thick as this? Normally, you don't get that. That's them being friendly to you in the exam. This person is heterozygous, and this person is homozygous dominant. Okay, again, I haven't even read the question yet, but this is the kind of process you need to do as you're going through the question. Like, that's obvious to me. Did it, like, I guess it's because I've done heaps and heaps of these. That I know I'm going to need, almost certain. Because this, it says here that this is, the little r is bigger, and so it moves less distance. Shorter fragments move further. The shorter one is 135. The shorter one is R. So the shorter one has gone further. Therefore, this must be the big one, this must be the big R, which is the smaller fragment. Okay, this is then a question. Explain, give reasons why primer A3 and primer A4 only bind to a specific DNA fragments. 2 marks. Two marks. How concise can we be here? They have a specific sequence of bases only complementary to a specific sequence of bases on the DNA, e.g. big R, or the sequence for big R or little r. We should maybe say the primer has a specific sequence of bases, which is only complementary to the specific sequence of bases you could maybe throw complementary base pairing in there as an insurance command word. Using the information, explain the results. Well, 3 marks. Why is it 3 marks? What do we need to say? Well, this is our answer that I've already tested you. Basically, the answer here is L equals little homozygous recessive, M equals heterozygous, and N equals homozygous dominant. That's why it's 3 marks. And again, things like this, when you've done enough past papers, this is immediately obvious. There's no doubt in my mind. I might expand upon this to link it back to the information. I wouldn't literally just write that, obviously, but these are the bits that we can say that this is the larger fragment, 195, moves less distance, because we need to explain, because that's our command word, but that's basically what we need to do. I'll share the mark scheme and see if I'm right. I haven't looked at this. Is an ancient paper. This is actually a reserve paper, which was never sat. Well, maybe it was sat. I don't know whether they did the reserve paper this year or not. The scientists wanted to know which chromosome the gene for the alleles R and R was located. The flies with the genotype homozygous dominant, they obtained cells that were in mitosis and labeled a DNA probe specific to allele R. They then looked at the cells under an optical microscope and explained why they used cells that were in mitosis. Very strong metaphase. Because the expanded, what can we say? The chromosomes have condensed. The chromosomes in interphase, and you look at the nucleus, you're going to see this. You're going to see there is no visible chromosomes. Whereas during the chromosomes are only visible after they've condensed. You could say visible have condensed. So otherwise it wouldn't be possible to say which, where, and then we'd maybe want to say, well, second mark, chromosomes are only visible in mitosis because they have condensed. What's the second mark? I'm kind of tight on time, so I'm just going to skip that and I'm going to show you the mark scheme before we go. Again, I'll rattle through this. Another group thought that the pesticide resistance in sunflies was related to increased activity of an enzyme called PM. This enzyme breaks down insecticides. Scientists say large numbers of resistant and non-resistant flies, then set up the following experiment. Non-resistant flies exposed to the insecticide, resistant fly exposed to the insecticide, resistant flies treated with an inhibitor, and then exposed to the insecticide. They determined the percentage of flies that were dead at different times after being exposed. Here are their results. Percentage dead, time after being exposed. We've got resistant flies, but there's still some of them die. Resistant flies with the inhibitor, and they still, okay, explain why the scientists carried out the control experiment on non-resistant flies. It's a control experiment, it's not that PCR related. And then the scientists concluded the resistance to insecticide is partly due to increased activity of the enzyme, but other factors are also involved. Explain, give reasons for how these data support this conclusion. You're not evaluating it, you're giving evidence of why it does support it for 4 marks. Well, basically, why are other things involved? Well, we can say that resistant flies are not 100% resistant, that there must be some other factor involved, that resistant flies with the inhibitor, what's the question? I need to get this question clearer in my head, is partly due to the increased activity. That the resistant flies with the inhibitor are resist dying better than the non-resistant flies. So there must be something else. These should be the same. If it was only PM, these should be equal, but it's not. Also, if these have functioning PM, then, and PM is the solution to the problem, then there should be basically 0% of the resistant flies, but they're not. So there's other things happening in this scenario. Question 9. Okay, so what were the answers? Why do you use the same restriction enzyme? To cut the DNA at the same recognition sequences. So you get fragments are the same. You extract the same bit of DNA. So I was along the right lines. This was why do the primers, why are primers A and primers B, I think they were called, specific? Each has a specific base sequence. Each primer, this was talking about, that is complementary to the allele. Obviously, primer A to the one allele, primer B to the other one. And then what was the answer to why? Explain the results. Fragments. Okay, so we don't get a mark for saying this. We have to say because it's an explain question. They were longer fragments, basically, was the reason why this one is 195, is because they're longer basins. And then we can exactly the same, but we can say that these are the shorter fragments for the big R's. And then we can say this is heterozygous because we've got 1 and 1. Why use two cells in mitosis? The chromosomes are visible, so we can see which chromosome the DNA probe is attached to. So that was the second mark in terms of that. Why? Okay, so for comparison, why do you choose them as the control? So whenever we're talking about controls, basically this is the golden little phrase to allow comparison between, and then you really want to state what you're comparing, the experimental groups in this case, to see the death rate in non-resistant, to see the effect of the insecticide in normal flies or non-resistant flies. So just to follow that up, to allow comparison between the two groups is basically your first sentence with why use a control, and then to see the effect of blah. I need to go and then that is the mark scheme for the other question, which I will leave you to follow up on the recording. Ronnie needs the room, so I will see you this time tomorrow. Bye.